Hello, I'm Angela Milton-Chelsea, a professor in the Department of Microbiology and Immunology. I'm your course director for microbiology throughout the pre-clerkship period, as well as your co-module director for the Multisystems and Complex Diseases module, which is the last one in the pre-clerkship period. You can reach me at the email address listed here, and just stop by my office in the B building, number 4096, if you ever want to talk. And I know some of you haven't had microbiology. This is just a brief introduction, particularly to medical microbiology, but I wanted to start with a shout out to Antony van Leeuwenhoek, who's considered the father of microbiology. He developed his own microscopes, and he ended up looking at things like red blood cells and protozoa and bacteria, spermatozoa under those microscopes. So really important for microbiology to be able to see these organisms or cells that we're looking at. And he was doing this in the 1600s, and I've just put an image here of a microscope from 1913. So you can see the development over a couple hundred years is really pretty amazing. So we're going to talk about pathogens today briefly, and these pathogens can be categorized into different types. So we'll talk about bacteria. Here's just a colorized image of salmonella, which causes diarrhea. We'll talk about fungi. So down here in the lower left, causative agent of histoplasmosis, which is a problem in the U.S. Southwest. We'll talk a lot about parasites. Just millions and millions of people around the world are infected with parasites. And this top one in the middle is Plasmodium. And you'll see these ring form and the schizons of the malaria. And you'll hear about that, particularly in multisystems. Throughout your pre-clerkship, you'll hear about other types of parasites. Here's just another picture of, this is a worm ascaris egg. Lots about viruses during the pre-clerkship period as well. And finally, just briefly, you'll hear about prions, mostly in the neurology module. Almost all the images from my recording are from the PHIL, the Public Health Image Library that's held at the CDC website, and they're very useful images in the public domain. So bacteria. Bacteria are single-cell prokaryotes, and as prokaryotes, they don't have a nucleus, so they don't have a nuclear membrane. Of course, they have chromosomes, usually circular. They don't have mitochondria, but of course, they make energy, ATP. They don't have Golgi bodies or endoplasmic reticulum, but of course they have ribosomes. They're making protein. They are growing organisms, but very tiny. We use our microscope, of course, to see them. They're 1 to 20 microns. They come in different shapes, generally cocci and rods. So our circles here are cocci. These are our rods here, but they also come in corkscrew shapes and some other different types of shapes. And you'll hear about that throughout the pre-clerkship period as well. Bacteria divide by binary fission. That is, they replicate their chromosome, and then the cell pinches in between those two chromosomes until it can divide into two daughter cells. We like to stain our bacteria so we can see them more easily under the microscope. So one of the major stains we use is the Gram stain. Bacteria with a thick cell wall will retain that Gram stain, and they'll stay purple or dark blue here. And if they don't have that thick cell wall, they cannot retain that stain, and so we counterstain them with a pink stain, and they'll be pink, so Gram negative. And this is important not just for looking at structure for microbiologists, but also as clinicians. They use antibiotics frequently that will target either Gram positive or Gram negative specifically. Fungi. So fungi are eukaryotic organisms. They have your nucleus, your mitochondria, your Golgi, and your endoplasmic reticulum, as you would expect. They can appear as yeasts or filamentous molds. They could be dimorphic, that is, they have both the yeast form and this filamentous form, and these can cause a variety of diseases. One that you've probably all heard of is athlete's foot, and this is the agent here. So parasites. Parasites are very complex eukaryotes. They can be unicellular or multicellular, and they can be really tiny protozoa up to really long tapeworms. And here's our Plasmodium again inside these red cells with that ring form there. Here's a hookworm. Schistosomes, you'll hear a lot about that. Schistosomes are endemic in a lot of parts of the world and really causes problems there. So viruses, of course, we're all familiar with viruses. We've all had numerous viral infections throughout our lives. These agents are really small. Again, they have nucleic acid, DNA or RNA. The DNA or RNA is either single or double-stranded, and they have one or the other, not both. And they also carry proteins along with them to help their replication. They are encased in a protein coat, and that protein coat may or may not have a lipid membrane. These viruses require host cells for replication, so can really be considered a type of parasite. They come in a number of different shapes. This bullet form over here is rabies virus. A lot of viruses have a more spherical shape, like HIV, influenza, SARS-CoV, and here's adenovirus with this really different shape here. And this last one over here, this is a virus that infects a bacteriophage. It's a classic image, but this is a virus that attacks bacteria, not humans. So prions, our last example, these are just proteins. These are proteins that can take a protein in your cell that's folded normally and cause it to misfold. This occurs in the brain. It could cause really bad problems. It can be either an inherited or an acquired disease. You can get it through contaminated tissue or meat. And again, it's a mutated or misfolded form of a host protein. So example over here, if this is the normal protein, kind of this open structure here, and then it's kind of folded up more tightly on itself in this on the far right here, this is can be disease causing over time. They have no virion structure, and they don't have a genome. As such, this folded tight protein, you can imagine it's resistant to chemical, heat, and ionizing radiation, so difficult to get rid of. So when we're talking about infectious diseases, there's a number of steps that have to occur in order for you to end up with a disease process. So you need to encounter that organism. If that organism isn't in your environment or somehow near you or acquired by you, you will not get that disease. For example, malaria. We don't have malaria in the United States except in travelers who come back from other countries. Very, very rarely can be transmitted in the US, but that organism is just not here, endemic in the environment. So you will not encounter it here in general, and so you won't acquire that disease. The organism has to enter in some way and spread and multiply, and then that multiplication or that adherence or some factor it produces causes damage, and the outcome would be a disease process. Remember too that host defenses are breached at each step by that organism. If the host defense can stop it, that disease will not occur, and you'll hear about that in a Ready, Set, Heal from Dr. Davies. So talking a little bit more about encountering, we can encounter organisms that come from outside our body, just contact with microbes in the environment. You can ingest those through contaminated food or water. You could inhale those or be exposed to those through the air or from objects. Bites are a big source of infection. Other humans can be a source, as well as animals. You can also get disease from microbes that are on or in your body from your normal microbiota. Really classic example, you get a cut, and that cut on your skin could be contaminated with your skin bacteria and become infected. Remember that you have a lot of body sites that are in direct contact with the exterior. So easy, eyes, for example, your mouth and your nose, your skin, but even your digestive tract. Basically, you're ingesting something that goes straight down into your digestive tract. It's exposed really to the outside. Genitals and urinary tract also exposed. So again, talking about entry, organisms can come in and they can kind of just hang out on your mucosa. So for example, the microbe that causes whooping cough or pertussis just hangs out there, produces toxin, and those cause damage and you get the disease. Another example, after ingestion, the microbe that causes cholera produces a toxin in the intestine that causes the diarrhea. So other organisms can penetrate that mucosa or the skin barrier and get into tissue. So examples of that, insect bite, if that mosquito is carrying Plasmodium, the agent of malaria, you could get malaria from that organism. Again, cuts and wounds, I already mentioned that, but cuts and wounds can get infected and then enter the bloodstream and then target another tissue. So, for example, they could go to the heart and you can get endocarditis. Organisms can also come into the body from blood transfusions or tissue transplantations, and examples of this have included HIV, rabies, and fungal infections. So for an organism to cause disease, it matters how many of those microbes you've been exposed to, as does a site of contact with the body. So, for example, Shigella, which causes diarrhea, you only need 10 organisms to get establish infection and cause disease from Shigella. Very, very infectious and easy to acquire. This is in contrast to the causative agent of cholera, Vibrio cholerae. This organism needs millions to cause disease. So just 10 organisms isn't enough. Both of these come into the intestine, but again, Shigella is much more virulent at lower bacterial numbers. Vibrio cholerae causes a lot of disease throughout the world and deaths throughout the world. So I don't want to diminish that, but you need more of those organisms. It has to reach a certain threshold to cause disease. Again, in the face of the host defenses. So this graphic is just to illustrate that. So you acquire an organism, there's an incubation period where it's trying to establish itself, multiply, and spread. If it's unable to do that, looking at the purple line, it could multiply for a little while, and then your host defenses could overcome that, or it's not able to multiply for some reason. You'll have an asymptomatic infection. It'll go away. In contrast, you could have an kind of out of control replication. You get disease, and if it goes on long enough or is bad enough, that can result in death. In the more usual example, the organism multiplies, we get some disease, and then our body starts to control that, and we get the numbers go down, and we get recovery and cure. Finally, the last example here is that we get some disease, the body starts to control that, and then something happens and there's a rebound in that organism, and this can occur cyclically, and you'll hear about some diseases that can work like that, such as malaria. So how do they cause damage? So again, entering into the body, they are colonizing, they're multiplying, they adhere and or invade. And just the tissue destruction is enough to cause damage and disease in many cases. In some cases, they're producing toxins. That toxin can act locally, or that toxin can get into the bloodstream and act at other sites. You'll hear about both examples during the pre-clerkship period. And finally, our own body's response, or the immunopathology, can cause the destruction in the tissues, and that's another way to get damage. So here's just an example of some cholera wards. We're in the 5th pandemic for cholera, and this causes really severe voluminous secretory diarrhea that can be just treated with replacement fluids. Just imagine, all these people could be treated with just replacement fluids. But this is caused by a toxin that acts locally in the intestines. So there are host factors that inhibit the establishment of infection. So in the saliva, we have antimicrobial peptides and molecules that can target microorganisms. We could have mechanical ways, so coughing can try to expel an organism. The pH of our stomach, a lot of microbes don't like that. That will kill them or reduce their numbers, really important. Another mechanical peristalsis within the intestine, trying to eliminate organisms that way. Really super important, microbiota established on our skin and all throughout our body, not in the internal organs, of course, but intestines, skin, genital tract, have their own normal microbiota that's trying to keep those pathogens out. And skin is a physical barrier for organisms, to keep organisms out of our body. And finally, shedding. We can shed, for example, virally infected cells as another way to try to get rid of that organism. So in summary, we've talked about classes of infectious agents. We've talked about how you can establish an infectious disease and what the outcome of that might be. And finally, a reminder that some body sites are sterile, like our internal organs, but others have normal microbiota, and that's protective. And we'll talk more about microbiota during the Foundations module. So that's it for today. I hope to see you soon.